Dear Professor Korkin, thank you very much for taking the time out of your Sunday to be a part of our podcast.
My pleasure. Thank you for having me.
You're very welcome. I know that the Saturday was spent, in having fun with the kids. How do weekends usually look like for you, especially now during the coronavirus pandemic? Are they busier than they used to be, or is it approximately the same?
They're definitely busier because there is just it's always good to have fun with the kids and the family, but there is also a growing number of sort of research projects, and on top of sort of our normal research activity, we now have things related to obviously to coronavirus, and we're trying to address it pretty much aligned with what we've been doing so far, regarding phosphathogen interactions. But obviously, the students are very excited about the opportunity to help and to get the new insights about the virus. I myself am very excited and feel it's a part of what the scientists are. First of all, to help the society to battle those kind of challenges. So it's more work, but it's I don't consider it as like heavy work. It's kind of entertaining for your brain, and it's also important. So combined, it doesn't feel like this extra burden. But my family certainly does feel so.
But I understand the perspective that you're taking. It's like, A, it's fun, B, it's useful. So it's like, that's the best possible, not definition, but the best possible adjectives that you could have for a certain line of work. What has been the funnest thing in regards to your work, be it like in terms of the research going on for the coronavirus, or just your work through bioinformatics in general?
Well, I think it's throughout my career, and it's just the science of bioinformatics is pretty new. It's pretty young, and so when I started doing bioinformatics, working on bioinformatics back in 2000, 2001, 2002. So this is when I was a PhD student. So I could see the transformation of this discipline from being more fun to being more useful and actually and sometimes critical in scientific discovery. And now I think we are sort of reaching a very good balance between being fun for the scientists. That means it's interesting, right? So it's engaging and being very important and useful for the society and other disciplines. And I think this is I would consider it to be the golden years of bioinformatics, where we are in a perfect symbiosis with life sciences, and we're there to help, and we're there to lead to some discoveries. To me, the most exciting part of this is the prediction part, right? So bioinformatics is properly designed to make predictions. So that means that we are able to provide some new insights that can be tested through the experiments, sure, but this very first, the fact of the discovery, right? That you get to see or get to find something that no one else was able to find before you. I think that to me is the most exciting part.
How did it feel then to be the first person to actually discover, to model a 3D model of the coronavirus? How was the feeling?
Well, I mean, so this is, and I have to give the full credit to other labs. I mean, so we were essentially, I wouldn't call it in the arms race, but there were in parallel, there were multiple labs working, on solving the structures of the viral protein, right? So we just happened to be, I mean, we treated it as the sort of as a hackathon. So that means that for the 2 weeks we worked, essentially with very little sleep, and the whole lab worked in as and I was happy to see that the whole lab worked as a well-oiled machine. And so we felt the need for us to provide this information. And I was actually surprised, because typically the experimental data comes in much later. When we talk about the structures of the proteins, or structure of the protein machines, right, the complexes, because if you look at this the viral proteins, they don't act in isolation, they actually act together. They interact with each other, they interact with the host proteins, and this is what creates the virus functioning, right? So this requires tremendous efforts from the experimental community, and that's one of the primary reasons why, when we see the genome, the first genome, or first several strains of the SARS-CoV-2 genome, we essentially also see an opportunity to provide this early prediction, so they can be helpful to the experimental scientists, helpful to actually to design the to obtain the experimental structures that typically are much more accurate. In our case, we've seen, and in the case of other people who applied similar methods, we've seen that the models actually that we developed were very close to the experimental structure. And I was surprised to see that the experiments came in much earlier than I would anticipate. And so, but yet we got this, several months to, half a year of cushion that we managed to provide with our models. And so, so it felt, so this work felt to me as a very important one. It's what we are trained to do. That's what we've been doing as a part of our research. So I feel that, I feel obliged to actually to do that.
I have a feeling, and this is just from my layman's point of view, is that the whole research thing in terms of the coronavirus, looking at it generally from the research on the virus itself to, now providing for the vaccines and distributing these same vaccines, was incredibly fast, actually, much faster than it used to be. I remember back in the day listening to, I don't know, I believe it was some sort of a podcast where it was said that the usual time period for creating a vaccine is usually 2 years, because it takes testing first, of course. I assume, not of course, but I assume on animal subjects, then larger and larger animals, in the end on the human beings. But in this case, it seems really the whole scientific community took a maximum out of itself and out of its own efforts to provide the best possible solution in the closest amount of time.
Yes, I completely agree, and that's something that that makes me really proud. To see that's sort of, to me, that's one of the biggest purposes of science, right? So to be able to respond to the challenges, to the global challenges, such as this pandemic, in the fastest manner, ahead of everyone else. And that's what I see, that's what is happening. And it's just so rewarding to see how the scientific community joins its forces together and works in extremely sort of efficient and self-organized manner. And I agree, we haven't seen anything like that before at this scale. It's just it gives me hope. Gives me hope that what the scientists do is actually is important. Is one of the most important things that in the society.
And these results, I mean, how do you feel that these results, not just the results, but the knowledge that the scientists, you included, have acquired throughout this period, but also in terms of, as you said, one of the basic components in regards to the lack of words, components of bioinformatics is predictability. How much do you believe we have learned in terms of predicting diseases like this and in terms of dealing with diseases like this in the future? Because, of course, there was, like, again, looking at it through the layman's point of view, we had similar crises beforehand. It seems like we could have been a lot more prepared than it was the case right now.
Well, it's an interesting question because the previous crises, right? So including the 2009 swine flu pandemic, they were different. I mean, really the comparable crisis that one should take a look to was, and obviously everyone is talking about this Spanish flu, right?
Oh yeah.
in terms of the scale, right? So in a coverage. But back then, I mean, the virology was in its infancy, right? So we haven't had, half of the current methods. I mean, we didn't have sequencing, which is now, so helpful. We are getting and this is again, this one, one of the unique aspects of this pandemic is that the amount of the data we're getting is really unprecedented. And the times, I mean, it's like, so a lot of groups and laboratories have the sequencing capacity, so they're using it. It's becoming like almost like a household tool for the majority of the experimental labs, and it's helping us to not only to learn about the virus, that's given, but also about us, right? So the host and their response to the virus and their susceptibility to the virus and the way the sort of the disease happens inside in different cells and tissues in different individuals in different populations, etc., right? So so we, in a way, this is the first of its kind, and we've yet to learn, not just about this virus, but we've yet to learn about the consequences of this virus, right? So what are the long-term effects of people who survived the infection? So there are a lot of new things. There are things that we can sort of bring in from our knowledge about SARS, the original one, about MERS, because these are the two close relatives that are also sort of very virulent and evolutionary, very close. But we also learn, we are able to learn about influenza pandemic. We are getting the information from different sources, and people have done work on other pandemics and epidemics, and our lab including, but there are a lot of new things about this virus at different levels, at the atomic level, how it's organized, at the the course of disease level, the fact that the it has such a long, asymptomatic period, and the fact that you have the, very distinct asymptomatic cases, it's very different. You typically don't have, such a prolonged symptomatic period, and that one of the most contributing factors, in my view, to the infection spread.
So do you feel like this incubation or the asymptomatic period is the thing that makes the research on this virus most challenging, or is it some other aspect?
It's definitely making the research most challenging. It makes the containment of this virus more challenging. And we, in our work, we've shown that that's actually one of the critical points, and that's something that would make the review or revisiting of the containment protocols, right? So things that so what to do and how to react when you have these outbreaks in the future, because I think everyone has been talking about the fact that it's one, but it's not the last one, right? So.
Yeah, but I mean, as you mentioned previously, like, is the first pandemic of its kind that can be researched via like modern methods, unlike the Spanish flu, which is actually kind of like, this is something that I would like us to actually talk about a little bit more in terms of like the Spanish flu. If I recall correctly, the Spanish flu had two waves in one way, shape, or form. Like the first wave wasn't so lethal. Whereas the second wave, it was like I believe it was like some sort of a major mutation that happened, was a lot more lethal, and it happened, I believe, from 1924 up until some era. I cannot remember correctly, but it targeted like the younger population. If I can remember correctly, it was like something between 20 to 40 years old. What is so? There's obviously a correlation between like how deadly and how easily spread the disease or the virus can get. What's the actual component of the virus and the environment itself in which the virus habitates that dictates both its rate of spread, like of contamination, and its mortality levels?
Right. So, as a matter of fact, so scientists consider actually three waves of Spanish flu. Is just, you're right that in many countries the second wave was actually much deadlier, and there is an ongoing research why that happened, right? So I think there are at least two components to that. I mean, there are obvious mutations to the virus, but there is also this, the psychological fatigue, right? So the people have been under, whatever quarantine or containment protocols for months, years, right? And so by the time the first wave went down and it felt like, okay, so we are all over, many people just disregarded the basic protection, the basic containment measures, and that would contribute to the second wave, and in many cases we see the same thing happening nowadays. Of course, it's not purely because of the psychological fatigue, or because of the I wouldn't call it seasonality event, seasonal events, but they are sort of the events that essentially align the fact that during the summer people go to vacation, on vacation, because in some countries there are some major holidays, like in the United States Thanksgiving is a big holiday, big family holiday, right? So that could be contributing to the spread of the virus, right? So all these things are, the critical components in the formula behind the, how quickly the virus spread and how deadly it is. Mostly how quickly the virus spread, because the deadliness of the virus, of course, depends on the mutations, right? So if you look at the sort of quantitative components of behind the infection, so typically there are three basic numbers that can be sort of quantified. It's so-called infectivity, virulence, and pathogenicity. So pathogenicity is essentially the sort of, it's an umbrella term which essentially means that so if you have X number of people exposed to the infection and Y number of people who, well, let's consider the simple definition, who died from the infection. So, Y to X, this is your pathogenicity, right? But then this can be split in two components, right? So you are exposed to an infection. So what are your chances to get this infection, right? So that's your infectivity. And so once you get the infection, once you are infected, what's your chance to succumb to it, right? And that's the virulence. And so while the first one, so both of them actually, they are essentially dependent on the molecular organization of genome, the specific mutations, and in specific proteins, etc. Right, but on top of that, right? So you also have some, social components, some societal components that contribute to how quickly the virus spreads, right? Or whether or not the person wears a mask will contribute to the infectivity, right? So you can be exposed to this, the same person will be exposed to the virus, but with the mask on, the chances are much lower to get the virus, right? So there are many different contributing factors to the functioning of this virus. We are trying to understand, we're trying to decouple all these factors and to understand them first in isolations, right? So by so what is the contributing effect to each of these factors, and then once we understand well enough, we put them together into sort of a unified epidemiological computational model, and try to see what try to forecast the outcome of the outbreak first, and then global scale pandemic.
But looking at it like at the basic biological structural level, what's the component that actually makes a virus more contagious and more deadly?
It's typically more than one component, right? So of course, when you talk about the the ability of the, on top of that, actually something that I completely forgot to mention is like, it's what makes the virus to jump, right? So from one species to another, because that's in the first place, that's what happens to the majority of the zoonotic viruses. They jump. In this case, we pretty much are more or less confident that SARS-CoV-2 jumped from the fruit bats. And there's some some people are talking about whether or not they have sort of the intermediate host, these civets, or maybe not, right? So this is an ongoing research, and people are trying to understand this sort of the nature. So that's on top of that, right? But then essentially everything boils down, if in very late terms, to ability of the virus to to get attached itself to one of the human receptors, right? So this is where the majority of the research is focused, has been focused now. So how the spike protein of SARS-CoV-2 interacts with the ACE2 receptor, human receptor, right? So this is the primary interaction. There is a lot of research going on, including understanding how the mutations in the spike protein affect the ACE2 receptor. But there are also could be some variations on the human side that could make this receptor more or less susceptible. As a matter of fact, if there's been a recent work where they actually looked at the interaction between the same spike protein and ACE2 receptor in bats, in fruit bats, and they saw that actually there is a very strong arm race happening between those two viruses, and that there are many different versions of ACE2 in the bats, and so essentially there are difference, there are mutations. If you compare the same ACE2 in different bats, you will see that they're different, and those mutations are the key to the way how well or how badly the ACE2 interacts with the spike protein, and so now this is the key component. It's not the only component. So there are, essentially, most of the components in the virus, they are there for, specific functions. And depending on how this function carries out, right, it could be a bottleneck, with respect to how efficient this virus is, right? Mm-hmm. So, so it's important to study this specific, protein and its specific interaction, but it's not sufficient, right? In order for us to understand the pathogenicity, which and essentially the virulence of this of the virus, it's a much more sort of complex question in my view, because this virus is multifaceted, right? So it can cause damage at different levels, different cells, different tissues, right? So, and in many cases, this would be tissue-specific damage, right? So it would, it will be dictated by the, cellular and tissue organization in the environment in which this virus currently sits. So so we've been working now. We're trying to our lab is trying to develop machine learning models that are able to predict the pathogenicity of the virus, and use this machine learning models then to get back to us to inform what are the most critical contributing factors to the pathogenicity. Mm. There was a work, an early work by Eugene Koonin and his lab. They tried to get the first insights into the pathogenicity of the coronaviruses in general. So they took into consideration SARS, MERS, and they compared it to other non-pathogenic strains of the virus, or actually not really strains, but the relatives of the virus. Some viruses from the same family, mm-hmm. And so it's a very it's a very new for bioinformatics question. And I anticipate a lot of work to be done very soon, simply because we now have the access to unprecedented amount of data that can be used for that.
So basically, the pathogenicity and all of the and the other two aspects of when looking at the virus are still, like in broader terms, really early in regards to the research stage. But one of the things that, of course, if you can answer, if you cannot, that really interests me, and it's a hypothesis that I've heard somewhere, I don't know where, it's like that there is a limit to how deadly a virus can get. Looking at it simply from, for the lack of better terms, evolutionary perspective, because if it's too deadly, then it will not have any more hosts to latch itself onto, and therefore it will perish. Do you think that's a viable hypothesis, or does it lack some things?
Well, I mean, it's a sort of a common a line of thinking, and we've seen examples to this, right? Especially like to this, hemorrhagic viruses, Nipah virus, things like that. In many cases, I mean, those viruses are so deadly that they would, wipe out villages before, the villagers are able to transmit it. But this is, I mean, we are talking about, these are, extremely deadly viruses. So we're talking about like, the mortality rates of, 70%, 80%, right? So these are, so we are very far from this, right? So there is this huge range between where we are and where we, and the mortality rate at which we can start seeing these effects, right? So we are still, I mean, so it's an ongoing debate, right? So what will happen to this virus? Will it become seasonal? Will it stay, as we know it? Well, will it become more pathogenic, right? And so the current answer, no one knows. And I've seen works where, obviously it's tempting to put parallels with what we already know and what we've seen, before. But in many cases, it's very difficult to put those parallels because, I mean, you may want to try to put a parallel with, flu and, pandemic versus seasonal. But it's the way the flu, emerges. It's very different. So we have this reassortment events between the species, and it's in a way it's a different mechanism on how the flu jumps from the species. And SARS and MERS, luckily for us, it was contained well before it became the pandemic. So so in a way we don't really have a lot of things to put, parallel, to get some inspiration from, and therefore, so I've seen works where people say that, well, it's actually at this stage, it could be just by chance that certain mutations could be introduced that make this, the pathogenicity of this virus much higher. Again, how higher that will be, if we jump to single percentage, right? So now we are a fraction of the percent. If we jump to single percentage, it's a pretty big jump, right? So but in terms of the mortality rate, that is sufficient to essentially to contain this virus so people die before they spread it. It's, I don't think, and luckily we're not there yet. And another thing that makes this virus, again, something that I've talked about, the thing that makes this virus, more sort of resilient to those effects is the fact that we have a pretty long course of the disease, right? So it's not like, you see in the Contagion movie, right? So in Contagion movie, it's actually one of my favorite movies, and this is something that I assign as the first assignment to my bioinformatics class. So students come and see this, because it's actually the first half of the movie is extremely close. I mean, it's the closest thing to the science that I've seen in the sort of in the fictional movies, right? So but there you see the the person who gets it, is dead within what, 48, 72 hours, right? So in our case, in the case of the little outcomes, and it typically happens on what, day 21, 20, 22, something like that, right? So it's a very long period, and during this period, and during the first week or 10 days, the person is contagious, that doesn't feel it, right? So we don't have this effect where you get the virus, you dead, and it's so quick that no one is is able to contact you, and the spread stops. So
okay, but you mentioned earlier in terms of like putting parallels, of course, ungrateful parallels nevertheless between, for example, the influenza viruses and like the viruses from the coronavirus family, SARS, MRSA, and now the COVID-19, the disease that goes through the coronavirus. And you mentioned the jump that it makes from an animal to human. How does the jump from an animal to a human happen in terms of viruses, and what's so different, for example, between the influenza virus and what is currently known about the coronavirus?
Well, I mean, in both cases, you have the jump from the species, the animal species, and in the case of influenza, right? So we have essentially two main sources of jumps, right? Swine, which is the attributed to the name swine flu, and birds, which attributes to the name of avian flu, right? And so the way it's, but if you look at the most of the influenza subtypes, right? So something that we refer to H1N1, swine flu, or H5N1, or H7 and 9. So all these are different subtypes of the flu. And if you look at the sources where these, the genes that contribute to the genome of each of this virus, where they're coming from all these species, right? So there's, there are some genes that are coming from swine. There are some genes that are coming from avian flu. There's some genes that are attributed, that are human, right? So, and it's a soup of these things, and depending what what is the proper amounts of genes and sources, we have different subtypes essentially, and so in case of in the case of SARS and SARS-CoV-2, we don't necessarily have this reassortment event, right? So we don't have this combination. Instead, it's considered to be a single jump, right? So it's the event where you due to some mutation, your virus is able to jump from one species to another.
What enables, I'm sorry, but what enables, what sort of a mutation on like the molecular, so to speak, cellular level enables that sort of a jump? Because from what I've heard and from what I've seen, like there is a ton of like really deadly diseases that happen only on animals, but that haven't simply made the jump towards humans. So what's the difference?
The difference is in the ability to bind the receptor, right? Mm-hmm. So the receptors is the key to these jumps, right? So and it's literally, we typically use this analogy of the key and the lock mechanism, where if you have a locks that are very similar, then the same key can open
Mm.
all of them, right? So, and in our body, we have many different proteins that can act as receptors, right? It's not a single one. And so different proteins have different susceptibility to be targeted. And that's, I mean, it's an ongoing research. There was this kind of controversial study back in, I think, 2014. So this is where there was this moratorium on flu research. So where the scientists try to design the avian flu genome, so it would jump from one species to another. I believe it from flu to from birds to another species, non-human species, right? So it still was pretty safe to carry out, but even that was raised a lot of questions, and I think it was sort of banned for over 6 months or so.
Okay.
So it's an ongoing research. We've yet to understand the full picture of what makes this thing jump. If we are able to do that, we are able to predict the future pandemics, essentially. And we were able to see that, okay, so this strain is probably critically close to the jump, and we need to pay close attention to this one. This strain is actually might be very deadly in this host species, but it's unlikely to jump to the human. And in the history, there've been things where we were essentially saved because of the inability of the virus to jump. So this very well, now it became very popular, but when I read it, it was less popular, let's put it in this way. So the hot zone book, right? So it's actually based on the real events of the of the potential Ebola spread in Washington D.C., near Washington, in the Washington D.C. suburbs, right? And so there was a deadly strain, the deadly strain in their host, which were the I think monkeys.
Yeah, monkeys. They introduced the monkeys, and like a lot of them had the disease, and it took them like, I don't know, several days for the scientists to figure out that this is Ebola, like in Washington.
But what was really scary is the mortality rate. I mean, this virus wiped out essentially the whole this group of monkeys that was they were brought into this facility, right? So had this virus jumped over, it would be a disaster, right? And as a matter of fact, if you read into the what happens, actually, I believe 2 or 3. I think one of the scientists and 2 of the people who handled the monkeys in that facility got the virus, but the virus was not. It was pretty much benign for. So they had a full recovery, right? But and that just shows you that there are so many components that need to be done right in terms of the virus, right? For the virus to be able to jump from one species to another, that's one, right? For the virus to be able to jump within the new species, right? So we saw examples with recent examples with the avian flu that was extremely deadly. I think it's like 50% mortality rate. It's huge, right? But it was able to jump only from birds to human. Right? So the last step was not there, right? So all these components need to be there. And when I say components, I mean the right mutations that make these things work. And luckily for us, we don't see that very often.
I mean, when you take all of those things into consideration, like both the things that are known that need to happen actually for the, first of all, of course, the jump from the animal to human, then, for the right mutations to happen in order for the virus to successfully spread itself, plus to preserve its pathogenicity throughout the humankind. And then taking into account the things that we do not know, I mean, it kind of serves as a laughing point. I wouldn't say as a laughing point, but I would say it serves as a real critique, to the more or less like the conspiracy theories that are going on about the coronavirus being made by human hand, engineered. I mean, in that line of thought, how far do you believe that the humankind is actually not far in terms of time, but in terms of knowledge? How far do you think that the humankind is actually far from being able to engineer a virus, whatever the engineer word might mean?
Well, I mean, so there've been work done. There has been work done in terms of in those lines of research, right? So first of all, I'm not a virologist, so so I have my knowledge based on the literature that I've read, and so I've known at least two works, but probably there are more where scientists engineered the virus to be to alter its function. Right. So one that I already mentioned. So this is the 2014 work that essentially triggered the NIH moratorium, which then was lifted. And this is the work on whether or not one can engineer a strain of the avian flu to jump from one species to another. There was actually an earlier work done on the on mousepox virus, right? So this is a relative to the smallpox virus, and where there were two groups. So one is an Australian group, and I think a year later there was a group from the at St. Louis, the Washington University at St. Louis, where they essentially what they did, they took a virus and they the viral genome, and they genetically engineered it by including into this viral genome the human gene called interleukin-4. And this is a gene that is essentially responsible. It's for it's our response to the viral invasion, right? And so by doing that, it's the virus is able to overwhelm the immune system very quickly, and essentially by doing so, it disables the immunity, right? So the smallpox vaccine has been one of the biggest scientific marvels in my view. So that's sort of a huge achievement, right? But what the scientists showed on the smallpox virus, that it's possible to engineer the virus to surpass this the immunity through through the vaccine, and so work like that, it's they're done in very controlled conditions, and I don't have any concerns about this, because it's heavily this kind of research is heavily regulated, right? But so that's, so people have been trying, and the main purpose of this work is essentially to understand the nature of the virus to prevent these things, right? So by understanding how can we surpass the current vaccines, we can actually develop the tool, and that will help us to design a better vaccine. By understanding the way how the virus jumps from one species to another, we can hopefully try to prevent these jumps and forecast these jumps, right? So I think in general the biological engineering is designed for good purposes, and when regulated by NIH, by other agencies, it should be safe. It should be safe. But going back to your original question, I don't think that the virus was engineered. There is a lot of evidence that there is a natural sort of source, fruit bats, and people were able to essentially trace the evolution of the virus, and we're still looking by getting newer and newer genomes, we're able to further trace the evolution of the virus and see what are the critical events and how they affect the virus and its spread.
Yeah, I mean, plus, especially in regards to the last point, to the spread part, I mean, there's been not a lot, but there's been some research showing that, the virus in and of itself had shown its ugly head in Italy, way before March or February. Way before February, I believe. It was in February that Italy went under lockdown the first time. But I believe to tracing some sort of wastewaters and things of that nature, they've been able to trace it all the way till November. And it just, I mean, in terms of the perspective, it just shows how much research there has to be done in order to really grasp a sort of an understanding of it.
Absolutely, I agree. I mean, the fact that the virus could potentially be circulating in November, before it was sort of officially detected and announced, I wouldn't be surprised. I mean, typically it requires some critical mass, or sometimes sort of a matter of chance of getting to learn about this virus, right? And again, in many cases, it has symptoms of a pneumonia, right? Which is something very, very sort of common in after flu, one of the common complications after the flu, right? So it's difficult to detect a new virus. It's still very, I consider it pretty good luck for the scientists to be able to detect it and to confirm it and to characterize it so quickly. But yeah, I wouldn't be surprised if the virus existed before. I mean, as a matter of fact, one of the most recent works where they did this sort of the evolutionary analysis, it suggests that the virus, I think it first emerged in human, in humans around November, I think late October, November, something like that. So.
Yeah, it just goes to show how much research has to be done. Basically, because we're approaching the 1 hour limit that we've set around for our podcast. First of all, thank you very much for the insights that you've given us. And secondly, in terms of the future research that you're set out to do, in terms of the coronavirus and scientific breakthroughs, not just in terms of the things found out about the virus, but about the whole system being better and more efficient as a result of it. what are the things that you're kind of looking forward to discovering and working on in the future?
Oh, so I mean, there are a lot of very exciting directions that we are trying to pursue. Some of them we are trying to pursue. So one of the things that we've been focusing on is trying to model, now going from the building blocks, which are the viral proteins that we have pretty good understanding. We're now trying to make the viral particle. And so to create the model of the viral particle, which is, it's like one of the computationally most difficult challenges that I've had in my career. But I think it's also a very important challenge due to the fact that these viruses are extremely, the structure of these viruses is very flexible, right? So it's very difficult to capture the viruses in high resolution using the current imaging methods, microscopy methods, cryo microscopy, tomography. So the modeling will be of a tremendous help with all that. And on top of that, once we have an atomic level, resolution level model, we will be able to study the actual functioning of this virus. So how the interaction between the two cells happens, right? So we're going, we're trying to reach this, new level where we looking at this not in terms of the components but whole structure, right? So another word where we are, trying to wrap up is we're trying to. I mean, that's a difference, so a part of the spectrum. We're trying now to use the artificial intelligence methods to accurately model the spread, the real-time spread of the virus, during an outbreak event, with the idea that we will try to understand the current virus and the most efficient containment measures that need to be done, and that will hopefully provide some guidelines to CDC, to the Center for Disease Control, and sort of similar agencies throughout the world. But also we are making ready, we're getting prepared for the future outbreaks, because it's not a question if, it's a question when. And there are, SARS is one of the candidates, but there are some other potential candidates for these, zoonotic jumps. Nipah is one of the sort of novel ones that is, scientists have been closely watched. But as we saw, avian flu could be actually a very, very deadly virus, especially if it finds its way to how to spread, between the humans, right? So all these things require our immediate reaction, right? So, and ideally, when responding to this new outbreak, we should be equipped with the tools, both experimental and computational, to essentially to design our actions to be the most efficient and in terms of the response to the new outbreak. So that's what we've been working on, and that's, I think, it should be helpful for in the future, once we are once we're done with that and we share it with the scientific community.
I mean, it's going to be helpful on a breakthrough level. I mean, it's like, as you were explaining it, one of the shows that I really enjoyed watching when I was younger, and one of which I actually watch up until this day on National Geographic, is like the air crash investigations. So it's basically like one of the predominant themes and the predominant topics that like whoever writes these episodes mentions in the end, is basically like, everybody interviewed always says, if it weren't for this accident, we wouldn't be able to make these modifications to the airplane and to the computational system and to the psychological review of the pilots. They basically say, it took this accident to happen in order for us to create a safer future for the flight, in terms of flight, in general. So I believe like this massive acceleration in terms of efficiency and the quality of it being done in terms of both the coronavirus research and everything else included within the pandemic itself, your research included, that's what I'm trying to say, will really, might not pave a safe road for the future, but a much safer one than it was up until like December last year.
No, I agree. I think we are definitely learning from this pandemic. We're definitely becoming better prepared as a society, more aware as a society, and as a scientific community. I think we're definitely improving our knowledge about the virus and the ways and the measures to contain it and to study it.
There's a lot of hope for the future, in other words.
Yes, by all means.
Thank you, Professor Korkin. It was a real pleasure, and I hope that you have this remaining of the Sunday spent in a really good time.
Thank you very much. It was my pleasure, likewise.